conference, and I'm honored to be part of this conference. So one of the biggest challenge in neuroscience has been to understand how a single cell that is expressing a particular genetic defect can give rise to a very complex behavior, which can be autistic in nature, and how evolutionarily the complex brain, which is shown here in the middle, is able to utilize all the genetic defects that are existing in this particular cell type and has precisely been able to produce a behavioral output. So my search for understanding this complex behavioral repertoire of autism or repetitive behavior led me to use a very reductionistic approach wherein I wanted to understand how a genetic mutation that is occurring at the genomic level can influence a very specific cell type which is integrated in a particular neuronal circuit, and how all these things combined together give rise to a very complex phenomenon like behavior. So this led to develop a mouse model or even follow an existing mouse model and understand how the specific mutation in a particular gene could attribute to such complex behavior. So in 2002, this gene called HOXB8 gene was discovered, and a loss of function mutation of this HOXB8 gene leads to a very specific behavioral output, which is excessive grooming in the animal, which starts as early as 2 months of age and continues all throughout the life. And in 2010, it was discovered that the gene was very specifically expressed in 40% of entire brain microglia, which is an immune cell of the brain. And that particular microglia was somehow contributing to the behavior. However, there was a black box that was missing. How HOXB8 gene that is being expressed in a particular cell type like glia, which is an immune cell, now create a complex behavioral phenomenon like grooming behavior. And I came in 2009 to understand the neural circuit basis of this particular behavior. So what we know about HOXB8 cell population is this HOXB8 cell lineage gives rise to brain microglia. Shown here is the representation of HOXB8 microglia that is expressed in the brain, and these cells co-localize with a very well-known marker of microglia, which is CD11B. They enter to the brain through caudate putamen as well as a region called CC as early as postnatal day 2 and invades the brain and colonizes the brain as the brain develops. So the loss of HOXB8 gene function leads to this potential loss of hair from the body of the animal because the animal is exhibiting this excessive grooming behavior. And because we observed that the origin of these microglial precursors actually originates from bone marrow, a classic experiment was designed where it was thought that how about replacing the mutant microglia or the unhealthy microglia precursors with a healthy microglial precursors using a bone marrow transplantation procedure. And in fact, in this experiment, surprisingly, what they observed was bone marrow transplantation was able to rescue the repetitive behavioral phenotype. And in a reverse experiment, they observed that if you use a mutant unhealthy microglial precursors from bone marrow and transplant to a healthy mouse, you can induce the behavior in a control mouse, as shown in the panel E. And you can quantify how much time the animal spends in grooming in bone marrow transplanted versus non-transplanted animals. And what you see here in the graph is that the mutant animal spends almost twice the amount of time in grooming all throughout the life as compared to a control mouse. However, bone marrow transplantation reduces that grooming, and even hair patches will not be detected after the bone marrow transplantation. So that was a huge discovery, as well as that was a big leap in the way how we were trying to understand. But nobody knew in 2010 what exactly happened in the brain that led to the behavioral phenotype reversal. So, even to conceive as to what happened to the brain which led to behavioral phenotype reversal, we need to understand everything about what's going on with that cell type, the microglia. So shown here is the early entry of HOXB8 microglia in the brain, and in the green you see the microglia that are non-HOXB8 population, and in the red you see the microglia of HOXB8 population. So as you see, early in the development, the HOXB8 microglia is about to enter into the brain. However, the non-HOXB8 microglia already is implanted in the brain, already seeded in the brain way before the HOXB8 population comes in. And there is a real physiological reason why nature has chosen two populations of microglia to the brain, and you will understand more as to what these functions are as I move forward. So what we are using in the lab is a two-color mouse model where we label the non-HOXB8 microglia with a specific marker called CX3CR1 GFP, and HOXB8 microglia with a fluorescence reporter called Rosa TD tomato. And we create a double color mouse. With this, we are able to detect these two cell populations in the brain. And shown here is the quantification of how microglial density increases as we move from postnatal day 4 all the way up to postnatal day 32, which is 1 month time from the day of the birth of the animal. So here is the quantification of brain sections demonstrating that as you move across the postnatal age, you see that the density of the microglia increases in the brain. And it beautifully falls in a bell-shaped form. And you cut the brain in coronal section or in the sagittal section, you are able to see the same increase in the microglial density at about P8 to P16. And we call that as a critical period window at which the microglial density increases in the brain. So what's the importance of this increase in the density of microglia in the brain? So shown here is broad distribution of HOXB8 microglia in the brain, where you see a lot of green cells and a lot of red cells and a lot of co-localization of yellow cells. What we have seen is there is broad distribution of HOXB8 microglia in the entire brain. However, the percentage of HOXB8 microglia in selected sectors of the brain regions are different. And if you look at very carefully in this critical period time window as to what is happening to these microglial projections, you see that microglia of HOXB8 origin exhibit a lot of morphological projections or morphological features which are distinct from the non-HOXB8 population, which is shown in green color. And here is the zoom-in section of a brain slice that shows you the complexity of the HOXB8 microglial projections during the critical period. And why would they exhibit these complex projections? And there is a particular reason. And in this slide, I'm showing you the quantification of how the projections of the HOXB8 microglia changes across pre-critical period, during critical period, and post-critical period. And in most of the cases, we have seen that HOXB8 microglia exhibit higher significant amount of filamentous projections as compared to non-HOXB8 microglia. So we use very fancy tools in the laboratory to study how the brain functions. And one of the fancy tools is called Two-Photon in vivo imaging. So here is a mouse that sits below a microscope, and you can create a cranial window, and you can really measure and watch the dynamics of how these cells are interacting in a very particular specific brain region. And shown in the panel B is a three-dimensional volume of 1.5 millimeter of the entire brain to understand how the distribution of HOXB8 microglia is occurring in the brain. And shown in the panel D is the dynamics of the microglial projections during critical period. So in this panel, I'm trying to demonstrate that the motility dynamics of the projections of HOXB8 population is different from the non-HOXB8 population. And this is just the increase and decrease of the lengths of the projections of individual microglia that is measured across the time. And here is a heat map that demonstrates the dynamics of the microglia. So everything that you are seeing in yellow color represents the low motility of the HOXB8 microglial projections, and whatever you see in the blue color represents high motility. And what you see is in the pre-critical period and during critical period, which is the most important time window when everything is occurring in the brain, the dynamics of the HOXB8 microglial projections is way more higher compared to the post-critical period. So what exactly is going on during the critical period? If you label neurons and synapses, what we see is the HOXB8 microglial projections indeed co-localizes with the neuronal surface. And all the little white spots that you are seeing on the red microglia represents the synapses to which these HOXB8 microglia are talking to. And in fact, if you label these postsynaptic proteins with a protein called postsynaptic density 95, you see that the HOXB8 microglial projections co-localizes way more higher compared to the non-HOXB8 population. So that tells that indeed there is a communication that is occurring between HOXB8 microglial projections as well as individual synapses. And here is one of the classic way of understanding what microglia are capable of doing when you ablate a particular region of the brain. So we use a two-photon ablation strategy to create a small hole in the brain and watch what microglia are capable of doing. And when you look at this movie, all what you see is, in response to the damage that we have created, HOXB8 microglial projections, which were silent before, now uses their bulbous structure to attack the neuronal apoptosis that is occurring in response to the damage that is created by the two-photon laser ablation. So we then further probed these microglia to understand what their electrophysiological characters are, and we detected that they express the membrane property similar to non-HOXB8 population. So that's not a big surprise. However, a big surprise came in their response to ATP. So when you expose HOXB8 microglia to 1 millimolar ATP, we saw that at negative potential, their current amplitudes were significantly lower as compared to non-HOXB8 population. However, if you look at their response to positive potentials, you don't see that change. So there is something that is characteristic of these HOXB8 microglia at the negative membrane potential as compared to the positive membrane potential. But it gives you an understanding that these two populations might be different. So just to give you a summary of what I talked just now, is we have HOXB8 lineage that labels 40% of the entire brain microglia. They form extreme complex morphological features, and they show altered projection dynamics. And they also co-localize with neurons and synapses, and they exhibit distinct physiological properties. So now the big question comes. We have understood a little bit about HOXB8 microglia. Now the question is, how do HOXB8 microglia indeed communicate with synapses and the neural circuit? Because that's the targeted question which will help us to bridge the way how HOXB8 microglia might be interacting with the neural circuit in order to control the behavior. So this question is a big question, and it almost took 10 years to address this question. Because in order to understand what microglia is doing to the neural circuit, you have to understand which neural circuit you need to target. And in order to understand which neural circuit you need to target, you have to use a very specific methodology. And we started with global resting fMRI analysis of the HOXB8 mutants. And in this poor little mouse will sit in a conditioning chamber in the hope of getting a chocolate drop at the end of its session, and it has to sit 1 hour still without altering its head movement. And within that conditioning period, as well as within 60 minutes of the mouse sitting in that chamber, we are capturing the fMRI signals from all parts of the brain. And with around 22 brain regions that we analyzed, the only brain region that turned out to be showing a lot of connectivity differences was the corticostriatal circuit. The very selective circuits of the corticostriatal regions, especially the connections that are coming from medial prefrontal cortex to dorsomedial striatum, was hyperconnected, while all the other connections within the corticostriatal circuit, as well as beyond the corticostriatal circuit, were hypoconnected. So that is in line with some of the autistic brain phenotypes that people have observed in human subjects. So when we probed this in more detail, we also observed that there are regions like orbitofrontal cortex which also show similar kind of connectivity to striatum. But in general, the frontal cortex to striatum is the actual connectivity circuit that is playing a very important role in controlling this repetitive behavior. So when we looked at the differences between males and females, we detected that the females showed indeed a more robust connectivity phenotype in these brain regions as compared to males. If you look at more control regions like thalamocortical circuit or thalamostriatal circuits in the mutants, you don't see such kind of hyperconnectivity, which brings us close towards a very targeted circuit on which we can now further increase our study on understanding how this circuit is working. So we used a classic way of the procedures like electron microscopy as well as Golgi staining to understand what is going on at the individual synapse level as well as the expansion or contraction of these synapses in response to HOXB8 mutation. And shown here is the quantification of hundreds of dendritic spines that occurs on the neurons. And we observed that in cortex, the dendritic spines were increasing significantly compared to wild type, but in striatum, it was going in opposite direction. So here is a model to explain the corticostriatal circuit defect, where there is a hyperconnectivity that is going on between medial prefrontal cortex and dorsomedial striatum, but hypoconnectedness in all the non-corticostriatal circuit. And there were also synaptic expansion in the mutants in cortex, however, synaptic contraction that is occurring in striatum. So we did a lot of electrophysiological approaches to understand if the long-term potentiation is different in the mutant versus controls, and we observed that indeed a specific type of LTP, which is called as CNQX insensitive form of LTP, was exaggerated in the HOXB8 mutant compared to wild type. And in fact, if you carry out anxiety tests on these animals, besides the grooming behavior, they also exhibit higher anxiety levels. So shown here is two behavioral tests. One is the plus maze test, and the second one is the open field test. In these two tests, you can measure the anxiety levels. So whenever you see a lot of activity that is going on in the closed chamber, that means the animal is anxious as compared to exploring the behavior in an open chamber. So in both these behavioral tests, we observed that they exhibit higher anxious levels. In fact, they exhibited even social behaviors. If you expose a wild-type animal, which is at the center, to an intruder animal, a wild-type animal would interact with the intruder animal with more frequency as compared to the knockout, which has less frequency to go and interact. So we tested one of the known drugs that reduces the anxiety levels, which is fluoxetine, which is an SSRI, and we observed that we can rescue the grooming behavior, anxiety behavior, as well as social behaviors to be rescued by this treatment, long-term treatment of around 5 weeks without affecting the non-grooming behavior. So that give you an understanding of there's a way to pharmacologically rescue this behavioral output. But still, this does not give us understanding of how HOXB8 microglia can be a direct causality link to the behavioral output. So I started looking into these brains more in a more focused and in more structured way, and observed that different parts of the corticostriatal circuit exhibit different density of the microglia, especially the HOXB8 population. And in fact, if you quantify, we observe that in mutant mice, the microglia density decreases, both in frontal cortex as well as dorsomedial striatum. And they also exhibit reduced morphological features, which tells us that their interaction towards the synapses or the neurons is somewhat reduced. So I'll take you through a journey to the HOXB8 mouse mutant brain, and you will observe the distribution of the HOXB8 microglia in the entire mutant brain, and you see a lot of changes what happens in this brain. So here is a two-photon in vivo imaging zooming through the frontal cortex and demonstrating the pockets of the brain region where that pocket is secluded from microglia as compared to other pockets of the brain region where there is high accumulation of HOXB8 microglia, which in fact tells that there are hyperconnectivity and hypoconnectivity to which HOXB8 microglia are sensitive. And the red cells that you see, the small dots, represents the monocyte, and the tree-like structures are the blood vessels. And those are the black holes where there is no microglia. And there is this pocket which exhibits a broad density of microglia. And these are the same parts of the circuit where we have detected hyper and hypoconnectedness. So that gives you an idea of how the distribution of microglia occurs in the brain in order to distribute themselves in a specific regions which shows hyperconnectivity versus hypoconnectivity. So here is a cartoon that shows you that we can target very specific neuronal population within the bed of the brain, and we can in fact manipulate the neurons while we can watch what happens to the microglia. So there are a lot of new technologies that are coming which will help us to probe individual cells at the brain level, and we can very easily understand how HOXB8 microglia or the non-HOXB8 population interacts with the neuronal circuit. So this slide just shows you that there is changes in the microglia neuronal interaction, and it is somewhat defective in the HOXB8 mutant versus the non-HOXB8 population. And in order to understand how HOXB8 microglia in fact responds to the physiological stimuli, I performed a classic experiment of uncaging neurotransmitter like glutamate and GABA, and I observed that in response to the glutamate uncaging, you get step-like responses of calcium transients in HOXB8 microglia. However, that was not present with GABA. That itself tells that there is something that is associated with glutamate to which the HOXB8 microglia are responding. And in fact, if you use a metabotropic glutamate receptor agonist, you can elicit the same kind of calcium transients. And now comes a little bit fancy experiment. So these are all done in the brain slices, and definitely one want to understand what is actually happening in the real brain when the animal is shifting its behavior from normal behavior to excessive grooming behavior. So we use a small camera which is as small as your nail, and this camera can be implanted on the brain. And while the animal shifts its behavior, we can capture calcium transients in HOXB8 microglia, which is on the order of 1 to 10 micron in size. And in fact, when we did this experiment, we were so excited to look at these results because we induced grooming behavior artificially by just spraying the water to the mouse that was not grooming. Now, all of a sudden, when the animal shifted its behavior from normal behavior to grooming behavior, we were able to elicit these calcium transients. Takes a second to get to the next slide. So in fact, you can do a correlation plot to identify whether all the activity of HOXB8 microglia is getting activated at the same time when the behavioral shift occurs or not. And this question led us to understand that indeed, when the repetitive behavior occurs in the mouse, all the microglia are activated at the same time and they elicit the calcium transient. And in fact, these calcium transients are very complex, and they come with different patterns of the activity. Some are fast, and some are slow, and some are, average activation and deactivation kinetics. But that tells you that even within the HOXB8 microglia population, not every microglia are acting in the same way, but they exhibit different complexity of the calcium transients. So this is the histological verification of HOXB8 microglia expressing this calcium sensor, which is able to elicit the calcium activity in the mouse. And all the little stars that you are seeing are the HOXB8 microglia that this small camera is capturing from the brain. And here comes another fancy experiment. This is called optogenetics, where we can manipulate individual cells. And in this case, we are using HOXB8 microglia as a population to target a light-sensitive protein called chalaneropsin. And now, with this approach, we can directly induce a behavior by activating any cell that you want, or deactivate the behavior by silencing the neural circuit or the microglia circuit. So in this case, I'm targeting HOXB8 microglia with the chalaneropsin, and in short time you are going to see the light that is entering into the brain, and as soon as the light hits the brain, light is going to stimulate the HOXB8 microglia, and the animal which is not doing any grooming behavior will start inducing the grooming behavior. This is not a mutant mouse model, but this is just a control mouse model. And now you see at around slightly after half a minute or to 1 minute, you start observing this classic phase 3 striatum-driven grooming behavior, which is phase 3. That's what we call in the circuit of in the whole repertoire of the grooming behavior, and it enters to the phase 4 of the grooming behavior. And once you stop the light, you lose that behavior. So that itself indicates that now we can target very specifically any population of the cell types, including microglia, which is probably the first time that we have seen that a non-glial, non-neuronal subtype like microglia is capable of talking to the neural circuit and inducing a complex behavior even in a controlled mouse. And the other part shows you the quantification of the effect that we have seen, that the stimulation induces increased grooming time compared to the non-stimulation condition. So you can induce any type of behavior using HOXB8 microglia as a tool, and shown here is anxiety behavior that is induced by targeting a very specific location in the brain called basolateral amygdala, which is a fear-inducing location of the brain. So then you can ask a question, can microglia do everything what a neuron can do? Why then microglia cannot generate action potential? And there's a very, it's a very classic question, and we don't know the answer yet, but it tells us that brain can utilize various mechanisms beyond neurons, beyond neuronal networks to produce the same effect, and this will give us a direct window to understand how the communication between an immune cell and the neural circuit can control a complex behavior like the autistic behavior. And shown here is another behavioral paradigm, which also is representing the anxiety behavior in the mouse in response to HOXB8 microglia stimulation in the basolateral amygdala. So we have the histological verification to demonstrate that we are indeed stimulating HOXB8 microglia below the cannula that we are implanting. So here is a proposed model for the repetitive behavior. So somehow HOXB8 microglia is releasing some chemicals like cytokines or chemokines, which they often do when there is an infection or when there is a damage. And these cytokines are acting on the cytokine receptors on the neuronal surface of the striatum as well as frontal cortex. And this communication signal somehow is translated to the motor behavioral output, and that behavioral output is what we are observing as a grooming behavior in the mouse, or the anxiety behavior. So finally, in the summary, the HOXB8 mutants exhibit corticostriatal circuit defect. We observed that the dendritic spine pre- and postsynaptic structures are altered in HOXB8 mutants. There is a change in the synaptic plasticity of the HOXB8 mutant compared to wild type. There are also defects in the way how microglia interacts with the neurons, especially with respect to HOXB8 microglia population. And they also are capable of eliciting calcium responses in response to the induction of the behavior. And now, optogenetic activation of HOXB8 microglia can induce a very complex behavior like the grooming behavior, and which is also a repetitive behavior similar to what we observe in autistic mouse models or even in autism. And activation of HOXB8 microglia can also lead to site-specific behavioral induction. So I would like to acknowledge Mario's lab as well as the entire lab members who helped me to get to this point. Jeff Anderson, Robert Mark, Peter West of University of Utah for helping with various types of experiment, Longjun Wu at Mayo Clinic, and all the institutes that have supported our research. And thanks for your attention.